Hello dear friends, welcome to the SWAYAM NPTEL course on food microbiology for safe and sustainable food systems. The first is introduction, and the concept we will be covering in this introduction part is the food microbiology, why study food microbiology, history and development of food microbiology, where from these microorganisms come and where their functions. The food microbiology, microorganisms are living entities of microscopic size and include bacteria, viruses, yeasts and molds. Together they are designated as fungi, algae and protozoa. For a long time, bacteria have been classified as prokaryotes, means cells without definite nuclei, and the fungi, algae and protozoa as eukaryotes, means cells with nuclei. Viruses do not have regular cell structures and are classified separately. In 1990s, this classification changed and will be briefly mentioned in some subsequent classes. Microorganisms are present everywhere on earth, including humans, animals, plants and other living creatures, soils, water and atmosphere, and they can multiply everywhere except in the atmosphere. Together, their numbers far exceed all other living cells on this planet. They were the first living cells to inhabit the earth more than 3 billion years ago, and since then have played important roles, many of which are beneficial to other living systems. Among the microorganisms, some molds, yeasts, bacteria and viruses have both desirable and undesirable roles in our food. In this class, importance of microorganisms in food, predominant microorganisms associated with food, sources from which they get in the food, and microbiological quality of food under normal conditions are presented in the following slides. Why study food microorganisms? The microbiology is the study of microorganisms. The microorganisms, also called microbes, are organisms that are too small to see without a microscope, as you might have guessed. Microorganisms include algae, bacteria, fungi, parasites made of protozoa and worms, and viruses. From the adjacent figure, we can see some perspectives of how the size of the microbe compares to other objects and organisms can visualize. You can see here the viruses are the smallest in size, ranging from 0.05 to 0.1 micrometer, and the mycoplasma, they are slightly bigger than the viruses and they are having size of 0.1 to 0.5 micrometer, whereas bacteria, they are having the size ranging from 1 to 10 micrometer, and yeasts are still bigger in size, they are having size from 3 to 10 micrometer, whereas the eukaryotic cells, they have still bigger size ranging from 10 to 100 micrometer, and the biggest in size which we can see even by the naked eyes, more than 100 micrometer to several meters, and rest others they can only be seen by light microscope. or by electron microscope. In the human body, there are over 10 trillion human cells and approximately 10 times that many 100 trillion bacteria. These bacteria share the food we eat, help us to digest it and recycle nutrients from our waste. Some of these bacteria protect us, but others can make us sick. That is pathogenic microorganisms. We have bacteria on our hands, in our mouth, our nose and in our gut. Whenever they find an inviting environment, bacteria will make themselves at home. As well as being home to trillions of bacteria, our bodies also house various viruses and fungi. The community of microbes in a particular environment is known as microbiomes. Now, important notes. Just as each of us has a distinct fingerprint, each person has a unique microbiome. Those who live in close contact with one another, such as parents and their children, will have more similar microbiomes, but they will not be identical. The makeup of our microbiomes is affected not only by the food we eat, but also the people with whom we come in contact, the environments we pass through and the environment the food itself has passed through, and it makes its way from farm to fork. The differences among people's microbiomes may explain why some people are more susceptible to disease than others and why people react differently to drug treatments. Although there are many microbes capable of causing human illness and food spoilage, there are even more that are beneficial and in fact essential to the health of our bodies and the environment. Now, the history and development of food microbiology. Except for a few sterile foods, all foods harbor one or more types of microorganisms. Some of them have desirable roles in food, such as in the production of naturally fermented food, whereas others cause food spoilage and foodborne diseases. To study the role of microorganisms in food and to control them when necessary, it is important to isolate them in pure culture and study their morphological, physiological, biochemical and genetic characteristics. Although it is extremely difficult to pinpoint the precise beginning of human awareness of the presence and role of microorganisms in foods, the available evidence indicates that this knowledge preceded the establishment of bacteriology or microbiology as a science. The era prior to the establishment of bacteriology as a science may be designated as the pre-scientific era. This era may be further divided into what has been called the food gathering period and the food production period. The former covers the time from human origin over 1 million years ago up to 8,000 years ago. During this period, humans were presumably carnivorous, with plant foods coming into their diet later in this period. It is also during this period that food were first cooked. The food producing period dates from about 8,000 to 10,000 years ago and of course includes the present time. It is presumed that the problems of spoilage and food poisoning were encountered early in this period. With the advent of prepared foods, the problems of disease transmission by foods and of faster spoilage caused by improper storage made them appearance. Spoilage of prepared foods apparently dates from around 6000 BC. The practice of making pottery was brought to Western Europe about 5000 BC from the Near East. The first boiler pots are thought to have originated in the Near East about 8000 years ago. The adjacent figure shows different types of pots which were designed in the earlier days. The arts of cereal cookery, brewing and food storage were either started about this time or stimulated by this new development. The first evidence of beer manufacture has been traced to ancient Babylonia as far back as 7000 BC. The Sumerians of about 3000 BC are believed to have been the first Greek livestock breeders and dairymen and were among the first to make butter. Salted meats, fish, fat, dried skins, wheat and barley are also known to have been associated with this culture. Milk, butter and cheese were used by the Egyptians as early as 3000 BC. Between 3000 BC and 1200 BC, the Jews used salt from the Dead Sea in the preservation of various foods. The Chinese and Greeks used salted fish in their diet, and the Greeks are credited with passing this practice on to the Romans, whose diet included pickled meats. Mummification and preservation of foods were related technologies that seem to have influenced each other's development. Wines are also have been prepared by the Assyrians by 3500 BC. Fermented sauces were prepared and consumed by the ancient Babylonians and the people of ancient China as far back as 1500 BC. Another method of food preservation that apparently arose during this time was the use of oils such as olive and sesame. Jensen has pointed out that the use of oils leads to high incidences of Staphylococcus food poisoning. The Romans excelled in the preservation of meats other than beef by around 1000 BC and are known to have used snow to pack prawns and other perishables according to Seneca. The practice of smoking meats as a form of preservation is presumed to have emerged sometimes during this period, as did the making of cheese and wines. It is doubtful whether people at this time understood the nature of these newly found preservation techniques. It is also doubtful whether the role of foods in the transmission of disease or the danger of eating meat from infected animals was recognized. Few advances were apparently made towards understanding the nature of food poisoning and food spoilage between the time of the birth of Christ and in 1100 AD. Ergot poisoning caused by Claviceps purpurea, a fungus that grows on rye and other grains, caused many deaths during the Middle Age. Over 40,000 deaths due to ergot poisoning were recorded in France alone in 943 AD. But it was not known that the toxin of this disease were produced by a fungus. Meat butchers are mentioned for the first time in 1156, and by 1248 the Swiss were concerned with marketable and non-marketable meats. In 1276 a compulsory slaughter and inspection order was issued for public use in Augsburg. Although people were aware of quality attributes in meats, by the 13th century it is doubtful whether there was any knowledge of the causal relationship between meat quality and microorganisms. Perhaps the first person to suggest the role of microorganisms in spoiling foods was A. Kircher, a monk who as early as 1658 examined decaying bodies, meat, milk and other substances and saw what he referred to as worms invisible to the naked eye. Kircher's description lacked precision, however, and his observations did not receive wide acceptance. In 1765, L. Spallanzani showed that beef broth that had been boiled for an hour and sealed remained sterile and did not spoil. Spallanzani performed this experiment to disprove the doctrine of the spontaneous generation of life. However, he did not convince the proponents of the theory because they believed that his treatment excluded oxygen, which they felt was vital to spontaneous generation. In 1837, Schwann showed that heated infusion remained sterile in the presence of air, which he supplied by passing it through heated coils into the infusion. Although both of these men demonstrated the idea of the heat preservation of foods, neither took advantage of his findings with respect to application. The same may be said of D. Papin and G. Leibniz, who hinted at the heat preservation of foods at the turn of the 18th century. The history of thermal canning necessitates a brief biography of Nicolas Appert, that is 1749 and 1841. This Frenchman worked in his father's wine cellar early on, and he and his two brothers established a brewery in 1778. In 1784, he opened a confectioner's store in Paris that was later transformed into a wholesale business. His discovery of food preservation process occurred between 1789 and 1793. He established a cannery in 1802 and exported his products to other countries. This, of course, was the beginning of canning as it is known and practiced today. This event occurred some 50 years before Louis Pasteur demonstrated the role of microorganisms in the spoilage of French wines, a development rise to the rediscovery of bacteria. A. Leeuwenhoek in the Netherlands had examined bacteria through a microscope and described them in 1683, but it is unlikely that Eppert was aware of this development, and Leeuwenhoek's report was not available in French. The first person to appreciate and understand the presence and role of microorganisms in food was Pasteur. In 1873, he showed that the souring of milk was caused by microorganisms, and about 1860 he used heat for the first time to destroy undesirable microorganisms in wine and beer. This process is now known as pasteurization. This is the brief history of development of microorganisms right from starting from the 1546. In that, the Girolamo Francesco, he actually early description of the contagion. After that, the Antony van Leeuwenhoek, he discovered the first discovery of the microscopes termed animalcules. The Anton van Leeuwenhoek first discovery of microorganisms, which he termed animalcules with the use of a magnifying lens. After that, the Edward Jenner, a country doctor, showed vaccinating with cowpox can protect smallpox, and after that it goes through the 1800s and then 1800s to the 1931 and then 1940 the Ernest Ruska Max Knoll, he constructed the electron microscope, and after that the study of microorganisms were more easy. The immunization, the treatment of precautions of infectious disease has always been a matter of interest. The best approach for combating infectious disease is prophylaxis, that is disease prevention. Louis Pasteur in 1880 isolated a germ responsible for chicken cholera. He arranged one demonstration to prove he really had isolated germ that can cause disease. He inoculated the isolated germ into the healthy chickens, but surprisingly it had not caused disease. The reason was it inoculated several weeks old culture which had lost its ability to cause disease, that is nowadays known as virulence. Surprisingly, it was found that these attenuated forms having decreased virulence could stimulate the system of body to neutralize the effect of virulent organisms. This resulted in developing the concept of immunization. In 1778, Edward Jenner, a British physician, recognized the immunization procedure for smallpox disease. He observed the milkmaids who developed cowpox pustules on their hands and arms after milking cows infected with cowpox never had smallpox in their lives, that is, they developed immunity for smallpox. He transferred pustules materials from the infected cow to the skin of human, who shortly displayed the typical sore and later on become immune to smallpox. When deliberately inoculated with smallpox germs, this was realized by Pasteur and applied the same principle for the prevention of anthrax, and it worked. He called this innovated culture vaccines. Vacca means cow, and the process as vaccination. We now know that success of genus experiment was due to the similarities in the physical and chemical structure between smallpox and cowpox viruses, which could produce identical specific blood substance antibodies in response to the presence of foreign agents. This figure differentiates the vaccination and immunization. Vaccination is the act of getting the vaccine to produce immunity to a specific infection or disease, whereas the immunization becoming immune to a specific infection or disease, typically through vaccination. Now, the cellular theory of immunity. Metchnikoff in 1884, according to this, phagocytic cells, phagine means to eat, present in the blood and tissues of most animals. These phagocytes are the body's most important line of defense against infection. Humoral theory. Paul Ehrlich. According to this, soluble substances are in the body fluid, for example, blood serum, are responsible for immunity. It was observed that cell-free serum of certain immune individual was lethal to the bacteria to which they were immune. The serum caused the bacteria to clump together or agglutinate. The serum caused the bacteria to clamp together or agglutinate. This controversy was resolved by Wright and Douglas, who showed that although antibody was not necessary for phagocytosis, it enhanced the rate and extent of phagocytosis. Joseph Lister, an English surgeon, used dilute solutions of carbolic acid to soak surgical dressings that kept the wounds free from microbial infection, and healing took place rapidly. Chemotherapy. Prevention of disease through immunological response has its own limitations. Some disease can strike the person many times because infection fails to induce immunity. However, many diseases can be cured by chemotherapy, that is, use of chemicals that selectively inhibit or kill the pathogens without killing the patient. The modern chemotherapy began with Eldridge's search for magic bullets in first decade of 20th century. The compound 606 was very effective against and commercially named as salvarsan. The first antibiotic compound, pyocyanin, a pigment-like substance produced by green pus-forming organisms, Bacillus pyoceaneus. Synonyms is Pseudomonas aeruginosa. The chance discovery of penicillin by Alexander Fleming in 1929 was a major breakthrough in the field of chemotherapy. Fleming was studying the antibacterial action of variety of substances, for example, tears, saliva, etc. Due to contamination, mold growth was there in the petri plates and there was no growth of bacteria around that. He was working with Staphylococcus. The mold was later identified as Penicillium notatum. The first human trial was made in 1941. For this, Fleming, Florey and Chen was given Nobel Prize. The applied microbiology. While Pasteur and Koch were working in clinical microbiology, E. C. Hansen opened the way to the study of industrial fermentation. He discovered yeast and bacteria for vinegar production, whereas the Adamides he discovered the bacteria for butter production. Where are they coming from, these microorganisms? Following Leeuwenhoek's discovery, although there were no bursts of activity, some scientific minds were curious to determine from where the animal cues observed to be present in many different objects were emanating. Society had just emerged from the Renaissance period and science known as experimental philosophy was in its infancy. The theory of spontaneous generation, that is the generation of some form of life from non-living objects, had many powerful followers among the educated and elite classes. Since the time of the Greeks, the emergence of maggots from dead bodies and spoiled flesh was thought to be due to spontaneous generation. However, approximately 1665, Redi disproved that theory by showing that the maggots in spoiled meat and fish could only appear if flies were allowed to contaminate them. This advocates of the spontaneous generation theory argued that the animal cues could not regenerate by themselves, that is biogenesis, but they were present in different things only through abiogenesis, that is the spontaneous generation. In 1749, Turbeville Needham showed that boiled meats and meat broth following storage in covered flask could have the presence of animal cues within a short time. This was used to prove the appearance of these animal cues by spontaneous generation. Lazzaro Spallanzani in 1765 showed that boiling that meat infusion in broth in a flask and sealing the flask immediately prevented the appearance of these microscopic organisms, thereby disproving Needham's theory. This was the time when Antony Laurent Lavoisier and his co-workers showed the need of oxygen for life. The believers of abiogenesis rejected Spallanzani's observation, suggesting that there was not enough vital force, that is oxygen, present in the sealed flask for animal cues to appear through spontaneous generation. Later, Schulze in 1830 by passing air through acid, and Theodore Swan in 1838 by passing air through red hot tubes, and Schroeder in 1854 by passing air through cotton showed that bacteria failed to appear in boiled meat infusion even in the presence of air. Finally, in 1861, Louis Pasteur demonstrated that in boiled infusion bacteria could grow only if the infusions were contaminated with bacteria carried by dust particles in air. His careful and controlled studies proved that bacteria were able to reproduce biogenesis and life could not originate by spontaneous generation, that is abiogenesis. John Tyndall in 1870 showed that boiled infusion could be stored in dust-free air in the box without microbial growth. What are the functions of these microorganisms? The involvement of invisible organisms in many diseases in humans was suspected as early as 13th century by Roger Bacon. In the 16th century, Girolamo Fracastoro of Verona suggested that many human diseases were transmitted from person to person by small creatures and also indicated by Kircher in 1658. In 1762, Plancis of Vienna suggested that different invisible organisms were responsible for different diseases. Theodore Swan in 1837 and Hermann Helmholtz 1843 proposed that putrefaction and fermentation were connected with the presence of the organisms derived from air. Finally, Pasteur in 1875 showed that wine fermentation from grapes and souring of wine were caused by microorganisms. He also proved that spoilage of meat and milk was associated with the growth of microorganisms. Later he showed the association of microorganisms with several diseases in humans, cattle and sheep, and he also developed vaccines against a few human and animal diseases caused by microorganisms including rabies. Robert Koch in Germany in 1880s and 1890s isolated pure cultures of bacteria responsible for anthrax, cholera and tuberculosis, which was later known as Bacillus anthracis, Vibrio cholera and Mycobacterium tuberculosis respectively. He also developed the famous Koch postulates to associate a specific bacterium as a causative agent for a specific disease, which is as follows. A specific organism must always be associated with the given disease. It must be isolated in pure form. When inoculated into a healthy susceptible host, it must produce the disease. The organism must be re-isolated in pure culture form from the experimentally infected host. In order to test the pathogenicity of an organism, Koch postulates have to be satisfied in toto. He was the teacher who taught his students how to handle microbes and in popularly known as father of bacteriological technique. Along with his associates, he also developed techniques of agar plating methods to isolate bacteria in pure culture and to determine microbial number in a sample, the Petri dish, by Petri in his laboratory. Staining methods for better microscopic observation of bacteria and the use of steam to sterilize materials to grow bacteria. With time, the importance of microorganisms in human and animal diseases, soil fertility, plant diseases, fermentation, food spoilage and foodborne diseases and other areas was recognized, and microbiology was developed as a specific discipline. These are some contributors in the field of microbiology. Thank you very much for today.